Welcome! Today we embark on a journey into the hidden world within us. It is a world of sentinels and soldiers, of intricate codes and coordinated attacks. This is the story of our immune system, the most sophisticated biological defense force ever known. For centuries, its workings were a profound mystery. We knew it protected us, but we did not understand how. We saw its failures in plagues and its overreactions in allergies, yet the mechanisms remained elusive. This narrative will explore how, piece by piece, science illuminated this inner universe, transforming medicine and saving countless lives. We will trace this revolution through the lens of the world's most prestigious scientific award, the Nobel Prize in Physiology or Medicine. Our story is not just about cells. It is about molecules. It is a human story. It is about the relentless curiosity of scientists who dared to ask fundamental questions about life and death. How does the body distinguish a friend from a foe? Why do we sometimes become immune to a disease that has sickened us once before? Why does this powerful system sometimes turn against the very body it is meant to protect, causing various fatal autoimmune complications such as type 1 diabetes and transplant rejection? You will have answers to all these questions. Please stick till the end of this fascinating story. Each Nobel Prize in this field marks a pivotal moment, a turning point where a locked door was finally opened. These discoveries, often born from decades of painstaking work, built upon one another to create the foundations of modern medicine as we know it today. Over the next 20 minutes, we will travel through more than a century of these landmark breakthroughs. We will start at the dawn of the 20th century. We will then move through the decades, witnessing the discovery of the cells that devour invaders, the mapping of the proteins that tag them for destruction, and the decoding of the genetic language that defines our biological identity. This chronological journey reveals a powerful truth. Scientific progress is a relay race, where one generation of discoveries hands the baton to the next, building a tower of knowledge, one Nobel at a time. This exploration will reveal how our bodies fight off everything from the common cold to deadly viruses. It will explain why our immune systems can sometimes turn against us. Most importantly, it will show how these fundamental insights have been translated into tangible therapies, from the first life-saving serums to the latest cancer immunotherapies. They have given us vaccines, organ transplants, and revolutionary drugs, rewriting the rules of what is possible in medicine and offering hope where there was once none. Our journey begins in 1901, the year the very first Nobel Prize in Physiology or Medicine was awarded. The recipient was Emil von Behring, a German physician who developed a serum therapy against diphtheria. His work was a beacon of hope in a dark time. By demonstrating that the blood of an immunized animal contained a mysterious substance that could cure the sick, he not only saved lives, but also gave birth to an entire field of study. This substance, which we now know as an antibody, was the first clue to a complex system of defense circulating within our blood. Von Behring's discovery marked the dawn of immunotherapy and set the stage for a century of investigation into our body's protective powers. From that first prize, the Nobel Committee has consistently recognized the giants of immunology, creating a trail of breadcrumbs that we can follow to understand our own biology. In 1908, the prize was shared by two scientists with conflicting ideas. Ilya Mechnikov, who championed the cellular theory of immunity starring voracious cells he called phagocytes, and Paul Ehrlich, who focused on the humoral theory of chemical antibodies in the blood. It turned out both were right. Together, their work revealed the dual nature of our immune response, a foundational concept that underpins all of modern immunology, from the design of vaccines to the development of sophisticated antibody-based drugs. As we move forward in time, the questions become more refined and the answers more astonishing. How can our bodies produce a seemingly infinite variety of antibodies to recognize any threat? In the 1970s, Rodney Porter and Gerald Edelman meticulously mapped the structure of these proteins, revealing an elegant Y-shaped molecule with a variable tip. This discovery was the key that unlocked the development of monoclonal antibodies. Today, these therapies are essential tools used to treat cancer, autoimmune disorders, and viral infections, a direct legacy of that structural insight. The story continues with the discovery of the immune system's internal language. Scientists uncovered the major histocompatibility complex, a set of genes that acts as a personal barcode on every cell, allowing the immune system to distinguish self from non-self. This breakthrough explained the persistent problem of organ transplant rejection and provided a framework for understanding autoimmune diseases. More recently, the focus has shifted to control. Discoveries of the brakes on our immune system, like CTLA-4 and PD-1, have led to checkpoint inhibitors, drugs that release those brakes, unleashing a powerful immune attack against cancer. This epic of discovery, spanning over 120 years, is a testament to the power of science to unravel life's greatest mysteries. To truly appreciate the birth of immunology, we must step back into a world defenseless against microscopic invaders. The late 19th century was an era of immense scientific progress, yet for most families, life was fragile, shadowed by infectious disease. Cities were growing, but sanitation was poor. Breeding grounds for bacteria, diphtheria, tetanus, tuberculosis, pneumonia. Doctors understood that these diseases were caused by germs. Louis Pasteur, Robert Koch. But knowing the enemy was not the same as being able to defeat it. The tools of modern medicine simply did not exist. The prevailing feeling was one of helplessness. When an epidemic swept through a town, the primary responses were quarantine and prayer. Physicians could offer comfort, manage symptoms, but they had no real cures. For a child suffering from diphtheria, the outlook was grim. A thick gray membrane would grow in the back of the throat, making breathing a desperate struggle, often leading to suffocation. The bacterium released a potent toxin that poisoned the body, causing heart failure or paralysis. The mortality rate was staggering, reaching up to 50% in some outbreaks. The concept of immunity itself was ancient. People had long observed that survivors of certain plagues, like smallpox, rarely contracted the disease a second time. This observation led to early risky practices like variolation and later Edward Jenner's much safer smallpox vaccine using cowpox. But why it worked was a complete black box. What was this mysterious protective force? Was it something in the blood? Was it a change in the body's tissues? The questions were profound, and the answers were hidden deep within our biology, waiting for a new generation of scientists to develop the methods and concepts needed to uncover them. Some believed that immunity was a property of the body's cells. Others argued that the key lay in the body's fluids, the humors. This fundamental disagreement set the stage for a great scientific drama. It was a race to not only save lives, but to define the very nature of our internal defense system. The first major breakthrough would come not from a grand, overarching theory, but from a practical, desperate attempt to stop the suffering caused by one specific disease, diphtheria. The first glimmers of a solution came from a German bacteriologist named Emil von Behring and his colleague in Robert Koch's laboratory, Shibasaburo Kitasato. They were not just studying bacteria, they were studying the toxins that bacteria produced. They hypothesized the body might create a substance to neutralize those toxins. They started by injecting sublethal doses of diphtheria or tetanus toxins into animals. Small, repeated doses led to survival and later resistance to lethal doses. That proved immunity existed. But the next step stunned everyone. Could protection be transferred from one animal to another? They drew blood from a guinea pig that had survived tetanus and let it separate to isolate the clear, yellowish serum. They injected a healthy, non-immune guinea pig with a lethal tetanus dose and simultaneously gave it serum from the survivor. The result was astonishing. the animal lived. The immune animal's serum acted as an antitoxin, a protective shield. This was the birth of serum therapy. He and colleagues applied the same idea to diphtheria. They immunized larger animals, sheep, horses, to make vast amounts of serum. On Christmas night 1891, a dying girl in a Berlin clinic received von Behring's serum. Doctors and parents watched anxiously. Within hours, her fever dropped and breathing eased. She fully recovered. News spread like wildfire. Medicine now had a weapon, not just hope. It proved a fundamental principle. Our blood holds specific, powerful molecules to fight infection. The mystery of immunity was beginning to unravel. The impact of serum therapy was immediate and profound. It transformed diphtheria from a near-certain death sentence into a treatable illness. When the first ever Nobel Prizes were awarded in 1901, the committee sought to honor a discovery that had conferred the greatest benefit on mankind. There was no more fitting candidate than Emil von Behring. He was awarded the inaugural Nobel Prize in Physiology or Medicine. The Nobel Prize cemented von Behring's legacy as the savior of children and validated his groundbreaking concept. His discovery gave birth to the entire field of humoral immunity, the idea that our protection resides in the fluids, or humors, of the body. The mysterious antitoxins he discovered were later given a more formal name, antibodies. Von Behring's work demonstrated what is now called passive immunity. This principle is still used today in treatments for rabies, snake bites, and certain viral exposures. However, the story was far from complete. Serum therapy was a powerful tool, but it also raised new questions. While von Behring and his followers championed the chemical-based humoral theory of immunity, another group of scientists was looking elsewhere for answers. They believed the real action was not in the serum, but in the cells. While von Behring was focused on the chemical defenses in the blood, a Russian zoologist named Ilya Mechnikov was observing something entirely different. He took a thorn from a tangerine tree and stuck it into one of the larvae. The next morning, he looked through his microscope and saw the thorn surrounded by a swarm of amoeba-like cells. He named them phagocytes, devouring cells. Mechnikov believed phagocytosis was the primary mechanism of immunity. Inflammation to him was a healthy battle between cellular defenders and pathogens. He proposed a cell-centric theory, phagocytes as the heroes, foot soldiers on the front lines. That directly challenged the humoral theory of von Behring and Paul Ehrlich. The debate became a major scientific rivalry. On the other side of the great debate was Paul Ehrlich. He envisioned cells as having various side chains or receptors on their surface. A toxin, he imagined, would fit perfectly into one of these side chains, like a key into a lock. The cell would then be stimulated to overproduce that specific side chain, which would break off and circulate in the blood as a free-floating antibody. But more research made it clear both theories described parts of a larger integrated system. In 1908, the Nobel Committee resolved the rivalry. They awarded the prize jointly to Ilya Mechnikov and Paul Ehrlich. By honoring both men, the committee recognized two major arms: Mechnikov's cellular immunity and Ehrlich's humoral immunity. By the mid-20th century, a colossal puzzle remained. How could the body produce such staggering antibody diversity? This near-infinite repertoire seemed to defy genetics. The problem of antibody diversity sparked intense debate and two competing theories. The breakthrough came from two scientists working independently: Rodney Porter in the United Kingdom, Gerald Edelman in the United States. Combining Porter and Edelman's work revealed a symmetrical, iconic Y-shaped antibody. This solved the diversity puzzle. Variable tips create unique antigen pockets, while the constant region keeps interactions with immune cells consistent. Porter and Edelman won the 1972 Nobel Prize for this structural map. The ultimate application of this knowledge was the creation of monoclonal antibodies. This leap from structural knowledge to therapeutic application has been one of the most impactful stories in modern medicine. While the antibody puzzle was being solved, another profound mystery remained. How did the immune system know what to attack and what to ignore? The clues, it turned out, lay in the frustrating field of organ transplantation. The recipient's body would mount a violent immune response against the donated organ, a process called rejection. Working with mice, a geneticist named George Snell conducted painstaking breeding experiments. At the same time, in Paris, a French immunologist named Jean Dosset realized he had found the human equivalent. He called these markers the human leukocyte antigens, HLA. This set of genes came to be known as the major histocompatibility complex, or MHC. These molecules act as tiny billboards on the surface of every cell, displaying fragments of proteins from within the cell. Together, the work of these three scientists revealed that the MHC was the body's master barcode system. George Snell, Jean Dosset, and Benacerraf were jointly awarded the 1980 Nobel Prize. Today, no organ or bone marrow transplant is done without HLA matching. How did the immune system's killer T cells, the soldiers responsible for destroying virus-infected cells, distinguish an infected cell from a healthy one? The answer came from an elegant experiment by Peter Doherty and Rolf Zinkernagel. To their surprise, nothing happened. The T cells ignored the virus-infected cells despite the same virus. This result was profound. They had to recognize both simultaneously. The T cell needed to see the foreign what, the viral peptide, and the familiar where, the self-MHC, in one glance. This concept, MHC restriction, was revolutionary. Their discovery won the 1996 Nobel Prize. It transformed understanding of viral immunity and vaccine design. But a fundamental question remained unanswered. How did the body first know it was under attack? In the 1990s, French biologist Jules Hofmann was investigating. He discovered a gene he called TOL. Across the Atlantic, American immunologist Bruce Beutler had discovered the first human TOL-like receptor, TLR. The final piece was the messenger. Ralph Steinman discovered the dendritic cell, the indispensable bridge between innate and adaptive immunity. Their collective findings, which underpin modern vaccine development and cancer immunotherapy, earned them the 2011 Nobel Prize. But how does the immune system know when to stop? There had to be off switches, brakes. In a lab in California, an immunologist named James Allison proposed a radical hypothesis. CTLA-4 was not an accelerator, but a brake. Meanwhile, in Japan, another immunologist named Tasuku Honjo had discovered a different protein, PD-1, a second independent brake on the T cell. Honjo showed that many tumor cells protect themselves by expressing the molecule that engages this PD-1 brake, effectively putting nearby T cells to sleep. Allison hypothesized that by blocking the brake, he could unleash a powerful anti-tumor response. This was a completely new paradigm for cancer therapy. Patients with late-stage, metastatic cancers were experiencing complete and lasting remissions. Drugs like Yervoy and Keytruda have become standard treatments, saving and extending hundreds of thousands of lives. In 2018, the Nobel Prize was jointly awarded to James Allison and Tasuku Honjo for this discovery. But does the body also have dedicated police officers? A specialized force whose sole job is to actively suppress rogue immune cells and prevent friendly fire? Groundbreaking work from three scientists, Shimon Sakaguchi, Fred Ramsdell, and Mary Brunkow, finally identified these crucial guardians, a discovery so fundamental that it is recognized with the 2025 Nobel Prize in Physiology or Medicine. Sakaguchi identified the cells and their function, while Ramsdell and Brunkow uncovered their genetic master regulator, FOXP3. He named them regulatory T cells, or Tregs, proving that the immune system had a dedicated branch for enforcing self-control and maintaining tolerance. This work opened up a new frontier in medicine, to either boost these cells to treat autoimmune disease or suppress them to enhance anti-cancer immunity. The 2025 Nobel Prize for Regulatory T cells brings our journey full circle. From passive serum therapy to targeted manipulation of regulatory cells, an ever-deepening understanding of immune complexity. Looking forward, the field of immunology continues to accelerate. The next Nobel Prize might honor discoveries in the immunology of aging, the microbiome, or next-generation vaccines. Furthermore, the era of personalized immunotherapy is just beginning, creating truly individualized treatments with unprecedented efficacy. Each Nobel Prize has not been an endpoint, but a doorway to a new set of questions and a new world of possibilities. The inner universe of our immune system, once a complete mystery, is now a landscape we can map, understand, and engineer for our own benefit. The discoveries of the past century have saved millions, and as science continues to illuminate this intricate biological dance, the cures of tomorrow are already taking shape in the laboratories of today, promising a healthier future for all of humanity. These Nobel-winning discoveries didn't just deepen our understanding of biology, they gave humanity vaccines, immune therapies, and the power to prevent disease. Yet at times, these life-saving tools have become entangled in politics or misused by those guided more by fear or rigid belief than by evidence. It's important to remember that science serves everyone. It transcends ideology, faith, and borders. The immune system itself doesn't care who we are, only that we survive. Let's honor these discoveries not by dividing over them, but by using them to heal, protect, and unite.